I'm here with Dr. Janice Chen. I am so excited. We have so many questions, so I am going to dive right in. Janice, let's start right at the beginning. What do you remember about your early introduction to science here in Utah? When do you remember wanting to pursue science as a career?
Yeah, so as was mentioned, I grew up here in Salt Lake City. I was actually born in Southern California, but we moved to Salt Lake because my dad was a PhD student at the University of Utah in pharmaceutical chemistry. So, I have grown to love Salt Lake. I think it's an incredible place to grow up. And also the early teachers that I had, all the way from kindergarten. Actually, before that, I was um, the privilege of being a part of a Head Start as well. So grew up. My parents immigrated from China about 30 years ago, and we had so much support through Head Start. Through other programs that allowed me to get a quality education from the start. And so I went to Lowell Elementary School while it was still around. Went through the ELP program, and then West High School. Go Panthers! So uh, when it was in West from middle school through high school. And I think there have been just incredible teachers along the way. I think the ones that really stick out to me, certainly my math teacher. She was a true, educator, and she cared so much, not just for the students, but also just teaching math. And I think that was really formative for me in terms of just really appreciating, a STEM as a career in general. I think the other big motivator, and I think what shaped a lot of my interest in STEM as well is just the environment, the friends that I had at school. I think we were all very curious. We all really enjoyed our science classes. We would go to the library together to do homework. So I think just the environment that you're in changes a lot about how you engage with subjects and engage with the excitement of learning different things. But because my dad was a scientist growing up, I also was exposed to the molecular biology lab early on. And, he taught me how to run PCRs when I was in elementary school. We put those gels on our refrigerator right next to the pictures of the family. So I was really proud at learning how to do PCR at a young age. But I would say my real formative experiences in terms of deciding that I really wanted to go deep into science really started in my undergraduate journey at Hopkins. And that's where I had professors who really were willing to take a bet on me as a young trainee and allow me to do more independent research. I think that's also a really important learning, is like giving people the opportunity to learn, to fail, to drive towards independence. I think without independence, you just never know what you're capable of.
Okay, so when I get home, I need to get my kid to the PCRs if I want him to be a scientist. That's what
Exactly.
we say. Okay,
Exactly.
on it. So you eventually ended up at the University of California, Berkeley, and into Jennifer Doudna's lab, which is incredible. What do you remember about getting accepted into that program? Like, I'm trying to think of where this was in her career. Did you know how important that was going to be?
Yeah. So after I finished my undergrad at Hopkins, I was, in a moment of trying to decide what to do next. And at Hopkins, almost everyone, well, a lot of my peers decided to go to medical school, and I was like, you know what, I'm going to just take a year off and actually spend a time just doing more research, because I had the opportunity to do some independent research as an undergrad. I really wanted to spend a little bit more focused time only doing that. So that's where I applied to. Actually, I just cold emailed a bunch of professors around the in the US to say, hey, are you willing to, do you have opportunities? Can I join? So the good thing about cold outreach is like you only need one person to respond. So don't be afraid to put your name out there. You only need one. And that was the case for me. I had maybe two people respond, one that I was really excited about, and they fortunately had an opportunity for me to join as a research associate. So I jumped at it. That was at Harvard Medical School, and that was in the lab of Dr. Pam Silver, who is one of the leaders of synthetic biology. And I mentioned that because that was one of the kind of points in my life that really pushed me towards this theme of engineering biology. And I think that all of that is what I think about today as well. But in Tam's lab, she had all kinds of crazy projects. I was engineering microbes to make biofuels, and there were projects where they're trying to generate other synthetic molecules. But fundamentally, we were doing gene editing, but there were many different tools and different ways of doing gene editing. In fact, this was in 2013, 2014. So there was maybe just a few graduate students in the lab actually using CRISPR. A lot of us were using a lot of other old school tools to modify genes, and some of these organisms were quite challenging to edit. So I think even CRISPR at that time would not have really worked. But that was when I first heard about CRISPR, and that's where especially the use of CRISPR in mammalian cells was a new frontier at the time. So from there, I was really, it was clear to me that I wanted to do a PhD and not go into medical school. And so that's when I started to apply to different graduate programs. Um, and when I got the interview at Berkeley, I was super excited because Berkeley to me encompassed all of the basic fundamental research, which is really what I wanted to spend the next couple years of my life dedicating towards. So I was, I think being in Pam's lab and seeing kind of the translational applications made me realize in order to make that seed change of improvement, it really had to go back to understand the fundamentals. And that's where I think Berkeley as an institution really, invests and focuses in those areas. And fortunately got invited to interview. I loved my time there. And then I got my acceptance pretty shortly after. And I actually just canceled the rest of my grad interviews because I knew at that point I wanted to go to Berkeley. That's his first step. And then at Berkeley, you do rotations with different professors. And I was lucky enough to get a rotation in Jennifer's lab. And as you mentioned, it really was the early days. I mean, this was 2014, so just a couple years after the famous, CRISPR Cas9 paper showing that you could develop this molecule into a programmable gene editor. That was published, yeah, just a couple years ago. And that was ultimately what led to Jennifer and Emmanuel winning the Nobel Prize in chemistry. But there were so many unanswered questions about how this molecule actually works. What are the ways and the reasons why the enzyme is able to cut, a perfectly matching gene sequence while avoiding something that has mismatches. And so those are the kinds of problems I was really excited about. And I would say in those early days, again, Jennifer's lab was really focused on the biochemistry and really not the translational aspect at all. And during, over the course of the 4 years of my PhD program, that center of gravity was slowly transitioning. But early days, yeah, we were running like radioactive gels. We were, looking at the binding affinity of the RNA to the protein. We were trying to understand, the kinetics of cutting DNA. We were looking at one big area of her lab was looking at the structure of these enzymes because we truly did not know, what they were doing, what they looked like, what were the domains involved. So it was really basic stuff. And I think understanding this enzyme at the atomic level just really gave me a deeper appreciation of how all these things actually come together to make what I believe is one of the most transformative technologies we have in biotech today.
Right. So you talk about at that time they were still kind of on the chemistry side, and then started working on translationalism. I don't know proper word there. But now her lab is known as probably, I don't think there's another singular scientific space that has created more biotechs. What do you remember about the culture and why you think that shift happened? What did you kind of learn about building biotechs in that time?
Yeah. I mean, one of the amazing things that Jennifer did as a mentor was really give her students and postdocs and trainees scientific freedom to try new things. And I think in that way, everyone has their own ideas that, we're all working on really innovative technologies and we all have different ideas of its possible application. And she really gave all of us the ability to take that, really play that story out and see how far you can take it. And so, she would hire amazing postdocs who came from, they had a neuroscience background or they came from, they were clinician that were interested in doing basic science. So they kind of saw the end user and the end product and had a vision of what that looked like and was able to tie it back. So, I think that like for me, again, the culture of the scientific freedom, but also really understanding the impact of how your technology can, be relevant to the bigger picture. That's something that Jennifer, I think, has really excelled at. And that's the reason why she's been a leader, right, in this space. She was one of the first to even put a moratorium on the field of gene editing for germline editing as an example, right? So, and then she's also just been a public speaker about what this technology is. I think now when I speak to people, everybody has heard of CRISPR. When I started in the lab, like, maybe a couple people heard of the term, but I think her ability to communicate science to a broader general public audience are things that I've also learned as well. And I think that type of communication and community building is something that, we all think a lot about. And for her, I think because she's started so many companies and Mammoth is only one of, several companies that she started, it's like they're all her kids, right? She wants everybody to succeed, and rising tide raises all boats. And so I think we all are, we're all friends in the industry, even though maybe our companies themselves might be, in some level competing with each other, but we all understand like the end goal is to be able to, cure disease, help patients. And so I think, Jennifer's been a true leader for all of us.
Yeah. So from that experience, you eventually made it to Mammoth Biosciences. Can you talk about how the company came together? Where kind of the spark for that was?
Yeah. So I think in the introduction, it was mentioned that one of the discoveries that we made in Jennifer's lab was the realization that some CRISPR enzymes could actually detect DNA sequences. Before that discovery, everyone was really focused on CRISPR as a gene editing tool based on as I mentioned, the 2012 paper. But this was an example of, again, where we really just asked the question, okay, CRISPR is actually a naturally occurring bacterial immune system. And so therefore, there's many different flavors of CRISPR systems. In fact, early on in collaboration with my now co-founder, Lucas Harrington, we worked together to try to ask this question of, okay, Cas9 is the famous enzyme that everyone has heard about, but the universe of CRISPR is really vast. And that's because of the evolutionary biology behind how CRISPR has really evolved, over the hundreds of millions of years. And so looking through that lens, we collaborated with other labs like Jill Banfield, who is one of the leaders in metagenomic, sequencing, and we realized just how rich this toolbox was. But the problem is, we all saw kind of on a computer what was out there, but no one was actually doing the work of saying, okay, we can take these sequences on a computer and then actually get them working in the lab. And so that was the hard work that we had started to do. There were other labs in the world on the East Coast as well, trying to do a similar thing. And so everyone was really trying to like can for the gold in this like, gold mine. That was really what it felt like. We were like in the Wild West and really understanding, what the heck all these enzymes were doing. And so, the discovery of CRISPR detecting DNA was one thing I'm really proud of. And that was the early spark of like, oh, there's a really clear, near term commercial application. And then the other big discovery that we made was that there are these tiny CRISPR enzymes, like, the third of the size of the original Cas9 protein, that everyone thought were just artifacts of evolution. And, there were review articles made that said, anything that doesn't have this bilobed structure is probably not functional. And so a lot of the dogma of the field at the time was that, these little tiny things were, but, they weren't really anything. But we actually said, well, okay, actually one of the biggest challenges in gene editing is actually delivering these big molecules into tissues and cells. And, delivery is imperfect, but there are, clinically proven delivery technologies out there, such as lipid nanoparticles, which, us, including many other companies have been working on as a delivery modality, but that just gets to the liver. And then there's a whole class of AAV viral vectors that actually can get to different tissues outside of the liver. And the challenge with AAV is that you can't really fit, the original Cas9 protein into the packaging constraints. And so we said, okay, look, if these are the delivery modalities that are proven in the clinic, how can we innovate on the fundamental CRISPR itself? And that's when we started to say, okay, well, let's just try to understand if these tiny little CRISPR systems actually work. So we went through that, the hard to effort of doing the purifying the systems, doing the early biochemical work, and identified this activity. And that was a really important breakthrough for the field to say, okay, this toolbox actually is even bigger than we thought. And to us at the time, we were sitting on all these inventions, and we said, look, there really is no better opportunity than to just do it ourselves, because we, at the end of the day, really understood the technology. Being in the Bay Area and the ecosystem there, and having Jennifer, as a mentor and a co-founder, helping us provide those early connection, meeting Trevor, who is our CEO, their co-founder from Stanford, with a shared vision of how do we actually not just become the next CRISPR company, but really think about this is a broad platform that's going to have, long-lasting applications across many areas. And so we really wanted to, be leaders in that space.
Mhm. And speaking of being a leader, that marked for you a change from kind of being on the bench to being in a leadership position, being in the C-suite. What was that like for you? How did you prepare?
Yeah, it's a interesting question because nothing prepares you for becoming a company. I can tell you that. But what I think the PhD does do, which I didn't appreciate at the time, is you're constantly trying to figure out how to make things work. And you're constantly dealing with failure. And I think both of those things help you build resilience. And I think resilience is one of the most important things for trying something new, whether it's a company or starting a new research area, whatever it may be. It's just, and when I speak to patients and their families, like the resilience that these people have, like, that it's infectious, right? And so you realize, okay, this is like a true privilege to be able to be in this role and make the impact. I think I'm a very impact driven person. And so whether it was doing, the research on the bench, the lab, and thinking about, okay, what are the opportunities, to now as a leader of a company thinking, okay, like this is, there's a real momentum behind the opportunities, and there's, a real need that we have, that exists, that our technology can actually solve. And then, of course, everything that goes along with it. Yeah, I think the ability to navigate uncertainty and learning how to not just deal with failure, but how do you learn from failure? I think that's something you learn as a scientist that I think applies to everything we do.
Mhm. We don't have time to go through every piece of Mammoth's history, but when you talk about resilience, I absolutely think of the journey that this company has gone through COVID. You've changed what you're doing several times. Can you talk a little bit about that on the company level, like how you navigated that and how you brought Mammoth to where it is today?
Yeah. No, when we started, we never said we are a diagnostics company or we're a therapeutics company. We said, look, we have what we believe is the broadest toolbox in CRISPR today. And we have something that solves a problem that the field has not been able to do, which is, yeah, both delivering to tissues outside the liver, and also this completely new discovery that, again, it is not just editing DNA, but it's actually detecting DNA. I think those were sort of the pillars of the company when we started. But at the end of the day, the real secret sauce and our core competency was the ability to identify new CRISPR systems, understand how they work, and then apply them to application, whether that was something that we were doing as the company or something that we would partner. So I think early on, the business model was never that, we would be everything all at once, but that there was different stages of time, in the company's journey where, there was the right moment for us to focus on diagnostics, and then it was the right moment for us to focus on therapeutics, and then it was the right moment for us to partner some of these technologies where, we didn't bring in the disease or biology expertise, but somebody else did, and that was the way that we were going to connect the dots, and then ultimately show that our platform was going to have real applications, and be able to address some of the severe diseases, that patients are suffering with. So I think from that standpoint, you're absolutely right. During COVID, it was really a crazy time, as we all remember. In fact, I had an early collaborator, with a physician scientist at UCSF, Charles Chu, and he actually gave me a phone call and said, "Hey, look, we have some of the first samples, COVID samples in California. Do you want to try to build a test around this?" Because as you recall, at the time there were no COVID tests, right? And so, how can you say no, right? And so we jumped on it, and we knew that our detector technology, was, the whole point is that it's a programmable detection system, and so we could program it towards, the COVID virus, and really quickly showed it in the first patient samples in California. We're able to, detect the virus in these samples, and that became clear to us that this was not just a idea, but something that we could actually do. Now, the challenge with COVID, as we all know, is that there was a tremendous amount of investment, and so when we went from there were no tests, all of a sudden the market was flooded with tests, right? And so that need was no longer there. And so while we were really, proud of the work that we did to get something from nothing, right, to, an EUA approval, actually we got 2 EUA approves. We were planning to launch this in laboratories to help them increase the capacity of testing, but the reality of the market was that need was no longer there. There were already products serving, and so we said, look, as a company and as a business, like that doesn't make sense for us to continue pushing on this particular element. And so we said, okay, look, we have something that's actually tremendously valuable, and we were getting a lot of inbound interest in trying this technology. Said, okay, well, actually, let's create a licensing program, right? While we're not going to be the product developers and we're not going to be able to solve all these problems, there are people, other companies and academic groups and researchers who want to do that. And so how can we be the enabler of the ecosystem? So that's something that we've really focused on in terms of diagnostics. Like, we want to enable others with this technology. Now, you know, as you also mentioned, like, therapeutics has also been something that now is the primary focus of the company. But I think because COVID was so top of mind for everybody, honestly, no one was even on the outside world realizing that we were sort of pushing along these new, ultra compact CRISPR systems in the background. And I can tell you that was really, really hard work. In fact, nobody at the time was able to get them to work. And so we were, again, as a company that is, this is the benefit and curse of a platform company. You've got, many shots on goal, but you have to pick pick your bets wisely. And, we certainly made the wrong bets at certain times, but I think because of our ability to react to uncertainty and understand like where was the real need, I think that's when we said, okay, like and we were starting to see really positive data from our ultra compact CRISPR systems. We said, okay, look, we actually have something that we believe is really valuable, to patients and really solve this unmet need. And that's where some of our early partnerships helped us crystallize like there's a real, value proposition to the systems that we've really worked hard on. And then realizing that actually, again, like having the ability to grow the company, being able to drive your own destiny, and maintaining our own internal pipeline was also critical to long-term success of the company. And so now, with all that brief history, we are actually moving our first lead program into the clinic later this year. We're heads down, finalizing the regulatory filings start clinical trials in a cardiometabolic indication. So it's been quite a transition for the company from early research platform to now really becoming a drug development therapeutics company.
Right. And I think about earlier in your journey, you realized nobody was going to do this for you. You formed the company. And then as you were in that company, you realized, well, some of these pieces could be better off in somebody else's
Yeah.
hands. So, really exciting that you're in the clinic. What is kind of like the next year look like for you? You mentioned that you're kind of heads down. When are we going to like really be hearing about some big milestones?
Yeah, so just to clear, we're not yet in the clinic. We are
Ready?
hopefully, fingers crossed, everything so far appears to be on track, be in the clinic by the end of this year. But of course, there's things outside of our control. The regulatory agency needs to agree with our, accept the IND. We're already starting, discussions with the clinical sites. We need to be able to enroll patients. So there's a lot of, steps, between even getting an IND accepted and then being able to dose the first patient. But that really is the thing that we're working towards is, getting to our first in human trial, because at the end of the day, even if you have a really great preclinical and non-clinical package, like you really don't know how your drug is going to work until you test it in humans. And that's why we have clinical trials. But yeah, I would say, we are our just center of gravity is really focused on that clinical development, building out the clinical team. Actually this morning I met with one of the KOLs, not in this particular indication, but another indication. And again, just like really understanding what the patients really need. And I think that's one learning that I've had too, is like you always need to talk to the end user, right? Whoever that may be, you need to understand what they need, because oftentimes at a company we get caught up in like, oh, this is what the investor wants, or oh, this is what the agency wants. But you realize that everybody has a different risk profile, right? The risk that a patient who has no other options is so different from a regulatory agency who has to stand before Congress if a drug doesn't work, right? Which has a different risk profile from an investor who, has to show returns to the shareholders. So I think the understanding like what are the incentives that are there, and how do you as a company, you have to sort of figure out how to satisfy all of those and sometimes conflicting requirements. And to me, that's the fun part, right? It's a big puzzle. And what I've realized is that nobody knows the answer, right? And so somebody just has to have the courage and the conviction and the data to support that, hey, with the incomplete information, this is our best opportunity.
How is your leadership and your role changing as Mammoth does head towards that clinical milestone?
Yeah. Like I said, I'm spending a lot more time of my time talking to patients and physicians, and it's extremely rewarding. Like I said, oftentimes you may get caught up in a day to day struggles or frustrations, and then you talk to someone who is dealing with a completely different set of challenges, and you realize just like it just takes your breath away, and I think those moments give us the purpose to say, look, like all of these minutia, some things matter and some things don't, and I think that has allowed me as a leader to say, really focus on the most important things and sort of let go of the things that are not as important. In terms of my role, I mean, it's really changed a lot over the years. As an early scientist founder, now to try and, everything related to trying to run the business and ensuring that everything we do is ultimately in service of patients. But also, like I said, I mean, you are managing it at the end of the day. Drug development is expensive. You need to find a way to finance it, right? And so then you've got to talk to investors and capital allocators, and you have to talk to also recruiting, right? Like the pipeline of scientists and other employees of the company, like I worry a lot about that pipeline as well, especially now when the market is rough. Like I worry that like people are forgetting about that pipeline. So I don't know. There's a lot of things that I think about in terms of leadership, not just at Mammoth, but leadership in the industry, and I think related to talking to patients, realizing like we have a really important responsibility to not overpromise like what this can be, but really try to, but also at the same time like show that like we are really working on this, like we care. I think sometimes the industry is made to seem like, oh, we're just here to make money, but it's like the people that I work with and people I talk to, I mean, they care so deeply about ensuring that we can provide solutions, and so I think being able to have that communication is something that everybody in the industry, I think we can all do a little bit better job as well.
Mm. Do you still get any time at the bench, or are you firmly?
I tried in the early days to sort of do both, and I realized I'm really bad at both. So I realized, look, I'm just going to focus on, being a leader. But, the reality is, one of my mentors told me, like, as a leader, like, you never delegate the details. Like, you can delegate the work, but don't delegate the details. And I feel that where, I'm still reviewing the data, but I'm not the one actually executing the experiment. So in a lot of ways, like, those are the most exciting times of my day when I get to review some of the day-to-day progress, the incremental progress that over time builds up.
Yeah. So you were back in Utah visiting. We were talking backstage that, the ecosystem here has really changed. There's now a lot of growth in the biotech industry. Do you wish some of that was available to you
Oh my
earlier?
gosh. It's incredible what BioHive has done, what, Altitude Labs, Recursion, everyone here. I mean, it's like when I started, yeah, I don't think there was really a biotech ecosystem. I mean, like I said, my exposure was through my dad. And if he were not a scientist, I don't know what kind of exposure I would have had to the biotech world. So I think it's just really amazing what everyone is doing here. And I'm privileged to be in the Bay Area where you just like go to a coffee shop and there's probably like five other biotech founders or AI, startups like trying to pitch to investors. But like, that's like this, the culture in San Francisco is it's its own thing. But I do believe strongly that these ecosystems can exist anywhere. There's nothing like really uniquely special about the Bay Area except that, there's a lot of people who, there's a lot of momentum. But the same thing I believe can be done in Salt Lake City. I think the same thing can be done in Baltimore. I talked to groups that, because I was at Hopkins, I think people are asking the same question. Why can't Baltimore become an ecosystem? We've got, the NIH, we've got Hopkins, we've got this, universities, like all the ingredients are there. I think that same thing exists here in Salt Lake. I think it requires leadership, it requires conviction, it requires a pipeline, right? All of these things, I think that all the seeds are being planted. So I'm super excited what Salt Lake will look like, 5, 10 years from now with the biotech ecosystem.
Yeah. With our last minute, what gives you the most optimism right now?
Um, just talking to patients. I mean, patients are fighting so hard for these therapies, and that makes me want to fight even more.
Awesome. All right. Well, thank you so much, Janice. This has been excellent. We don't have time for a Q&A, but Janice is going to hang around. So feel free to ask her your questions off stage. But
Thanks so much, Annalee. Thank you.